Good morning, students. Today we'll just continue the lesson with what we stopped the other day. Let's move on to the next part. It's going to be about tiers of monocot stem. Okay. Now have a look at the picture that is being given for you. Here you can see the monocot stem diagram. An enlarged view is being given for you. There you can see, just compare the diagram with the previous task which we have done. We learned about dicot stem. Here it is monocot stem. Okay, have a clear idea. Here you can see bundles of monocot stem here. Okay, all three bundles are being given for you. Okay, have a look at the clear view is being given for you. Let's study how it looks like. Okay, of course the outermost layer, just like dicot stem, here also the outermost layer is going to be a single layer of epidermis cell. Okay, the other day I told you epi means the outermost layer, endo means innermost layer. Okay, this is again an epidermis which is going to be a single layer of parenchyma cells. Okay, it is covered with a thick cuticle. What is this thick cuticle? Cuticle is going to be the outermost covering, outer to that of the epidermis. Okay, just a thin layer only, one covering like structure. Okay, here stomata is present, but it is very few in number. Okay, let's go on with the next one, which is going to be the second layer. If you see, it's going to be the hypodermis layer. Hypodermis layer is made up of sclerenchyma. Here and there along with sclerenchyma you can see the chlorenchyma also. I repeat, sclerenchyma along with chlorenchyma. What is the use of chlorenchyma? Chlorenchyma means, of course we all know, the word itself is a chlor means it's going to be chloroplast. Okay, chloroplast is a photosynthetic pigments are there, so hence photosynthesis can be carried out in this area. Not much to say, very less. Just imagine, put in your imagination. Here you can have what is that, banana as an example here, then sugarcane as an example. All these things comes under monocot. The stem is all green in color. Okay, so stem layer, outer layer, which is going to be having a little amount of photosynthesis is being carried, not much to say, just a little amount is being carried here. The role of sclerenchyma is to provide mechanical support. Okay, the toughness of the plant is being provided by the sclerenchyma which is present in the hypodermis layer. The next is going to be the ground tissue. Okay, the ground tissue is going to be the next layer where you can see the layer which is going to be having the parenchyma cells. Here you can see a lot of parenchyma cells. Okay, I see circular structures around. Okay, the circular structures is going to be the hypodermis layer. Okay, it is just going to have the ground tissue. Let's see what is going to be there. There it has got a layer, a single layer of endodermis, cortex, pericycle and pith. Okay, the next is going to be the vascular bundle layer. In the vascular bundle you can see, here in this diagram you can see three bundles. Okay, in the previous diagram just only one was exposed for you. Here three bundles you can see. Okay, have a look at this vascular bundle alone. Here xylem and phloem is totally clearly differentiated in such a way you can see phloem in circles. Okay, here have a look at the diagram. Here you can see the phloem here. Are you all able to see, children? Okay, have a look at this phloem, then followed by the metaxylem and then the protoxylem. Okay, here the vascular bundles are all conjoined, collateral, endark and closed. Let's see what are these terms referred to. I remember telling you closed means there is no cambium. Okay, only xylem and phloem is present. Since no cambium is present, it is called as closed. Endark means you have got the protoxylem facing the center and the metaxylem facing the periphery. Such a condition is called as endark. And the term collateral, this and all we learnt in the previous classes. Okay, xylem lies towards the center and phloem lies towards the periphery. Such a condition is called as collateral. Conjoined means xylem and phloem are present on the same radius. So I repeat now, vascular bundle is going to be conjoined, collateral, endark and closed. This is very important, children, because this alone falls for a 2-mark question. A comparative question of the vascular bundles can be asked for you. So have this in your mind and learn this one. Okay. Next, a word about bundle sheath. Let's see what is this bundle sheath, a new word which you are going to learn here, the bundle sheath. Let's see what is this bundle sheath. In the bundle sheath is nothing but each vascular bundle is surrounded by a layer of sclerenchyma cells. Okay, if the vascular bundle is surrounded by sclerenchyma, such a condition is called as bundle sheath. This is an area where it acts as a storage area. Let's see how the xylem and phloem is going to look like. Okay, xylem and phloem is arranged in such a way, you can see it looks like a skull. What is a skull? Have a look. You can see two eyes, a nose and a mouth like structure giving an appearance like a skull. So we call it as a skull diagram also. Okay, basically here in the xylem you can see metaxylem and the protoxylem also. Have a look at the diagram again. You can see the largest cell which is going to be the metaxylem and the smaller one which is going to be the protoxylem. Okay, one term, new term gets added here called as protoxylem lacuna. In mature vascular bundle, okay, in the mature vascular bundle, the lowermost protoxylem disintegrates. You can see they all get destroyed and it forms a cavity. Such a condition is called as protoxylem lacuna. Okay, this is an important 2-mark question. You need to mark it in your textbook. Next is going to be the phloem. Here in the case of phloem, phloem parenchyma and phloem fibers are absent, whereas all the other structures like companion cells, sieve tubes, sieve elements, everything is present in the parenchyma. So have a look at this diagram. Okay, I repeat once again. You can have a look at this picture very clearly. What is given for you? Have a look at the notes also given, children, for you. Here are given pith is there which is going to be present and it stores the food. Here you can see the all the other parts also which is being given for you, where you can see the vascular bundle which is conjoined, collateral, endarc and open. Pericycle, it is present below the endodermis. Alternate patches of parenchyma and sclerenchyma is present. Okay. Here then you can have the notes also. Epidermis is given, a single layer with thick cuticle. Hypodermis made up of sclerenchyma and chlorenchyma. The ground tissue which consists of endodermis, cortex, pericycle and pith. The vascular bundle which is skull shaped, an important word. It is conjoined, collateral, endarc and closed. Okay. Next, regarding the xylem, it is V shaped or Y shaped. And here protoxylem lacuna is again a 2-mark question. Next comes the phloem. Phloem parenchyma and phloem fibers are absent. Pith is also there which is not differentiated in the monocot stem. Okay. Okay, a clear table difference between dicot and monocot stem is given. Okay, this is a very important 5-mark question. So I want all of you to learn this difference between. The first point is given regarding the tissues that are present, and the next tabular column is going to be dicot stem and the next one is going to be about the monocot stem. Have a look at this one. Regarding the hypodermis is given. In the dicot stem it is collenchymatous, in monocot stem it is sclerenchymatous. Ground tissue, here it is differentiated into cortex, endodermis, pericycle and pith, whereas in the monocot stem it remains undifferentiated. Next, vascular bundle. In the case of dicot stem, it is less in number, uniform in size, arranged in a ring, open and bundle sheath is absent. Whereas in the case of monocot stem, it is numerous, smaller near the periphery, bigger in the center, scattered, closed, bundle sheath is present. Next, have a look at the secondary growth. In the case of dicot stem, secondary growth is present, whereas in the case of monocot stem, it is absent. Next comes the pith. Pith is present in dicot stem and absent in monocot stem. Medullary rays, it is present in dicot stem, again it is absent in monocot stem. It's an important 5-mark question, children. You need to learn this one. Learn and write this one as a self-test so that it will become a practice for you. In this lesson, you can see lots and lots of difference between, okay, many difference between are present in this lesson, and questions at least one is expected to come from this lesson. So I want you all to learn and write all the difference between. I have given in your slides. You can learn from that one. Learn and take it as a self test. Correct yourself so that it will be practice for you. It will help you in future. At least one question will fall for you in your further exams. Okay. Let's go on with the next one. So far we have learnt about the root, the stem, and now it's going to be the leaf. Have a look at the picture of the leaf. Okay, all this time we were learning about the section that was taken for the root and the stem. Okay, now we are going to learn about the leaf. So you have to put in your imagination whereby when they take the section under the microscope, the root and the stem was cylindrical. Okay, what do you mean by cylindrical? Just imagine a pen like or whatever it is, okay, a round shaped structure where they take it as a section of it. Whereas in the case of the leaf, it is not cylindrical but it is flat in nature. Okay, so they take the sections in a cross manner. So we give the term as transverse section. Here again a comparison is being done between the dicot leaf and the monocot leaf. Let's see what is this going to be. Okay, let's study how the dicot leaf is going to look like and how the monocot leaf is going to look like. All this time I have been giving you a comparison between dicot and monocot. If you see, I have taught you about the dicot root, dicot stem, monocot stem, dicot leaf and the monocot leaf. Okay, I am going to go on with the leaf part. Now just recollect what you have learned in your lower classes. What is this dicot mean? What is a monocot mean? Okay, what is the difference between dicot? What is the difference between monocot? Just recollect yourself, children. You are learning in your lower classes regarding that one. Okay, a simple example which I can state here is all dicots will have a double seed and all the monocots will have a single seed. Okay, that is for your understanding which you need to learn. Okay, for dicot, peas, beans all comes as an example, and for monocot I can give rice as an example, paddy is an example, wheat as an example, where they have only one single seed. You can't divide that seed. Okay, they all come under monocot. Okay, again let's get back to the lesson where we've been learning with the dicot leaf. Okay, outermost layer, if you see here, you can see the epidermis is in two layers. Okay, here you can see the epidermis which is going to be on both the sides. Okay, upper epidermis and the lower epidermis. Outer to the epidermis, you can see one thin layer that is going to be the cuticle layer, which is a waxy layer. What do you mean by this waxy layer? Sometimes I will give a simple example for you. In the case of lotus, you pour water onto the surface of the lotus. What will happen? The water remains floating. It will be just like a mercury, it goes on floating. It will roll and roll and roll. It will not be touching. It is because of that waxy covering which is going to be the cuticle. Okay. Depending upon the cuticle layer only, the leaves get their protection. In some leaves it is thick in nature, some leaves it is very thin in nature. Okay, it offers protection, nothing else. Okay, let's talk about this upper epidermis. It is an outermost layer, and you can see one single layer. Okay, are you able to see one single layer? And here the diagram is given in green in color. What does it mean? The entire part of the leaf is green. That's a meaning to say about the photosynthesis and presence of chloroplasts. Okay, here in this upper epidermis, it is a single layer of parenchyma cells without intercellular spaces. Have a look at the diagram. There is no space at all in the middle. Okay, so it is continuous. I repeat the word, no intercellular space. No means nothing, inter means in between the cells there is no space, so we call it as no intercellular spaces. Okay, and here you can see the stomata which is going to be less in number. What is the role of stomata? It helps in the process of transpiration whereby gaseous exchange takes place, but in the case of upper epidermis it is less in number. Next, going on with the lower epidermis. Have a look at the lower epidermis. It is going to be the lowermost part. This again is going to be a single layer, but here it contains numerous stomata. In the diagram you can see a gap is there. Okay, are you able to see? A gap is there whereby you can see the stomata present, whereby what happens there? In that place only you can see the exchange of gases taking place. Okay, chloroplast is also present. It is present on the guard cells. Guard cells again, it is present in the stomata. 9th standard you would have learned about the shape and structure of stomata, which helps in the gaseous exchange, whereby you can see the oxygen going out and the carbon dioxide coming inside, that is present in the lower epidermis of the leaf. Okay, the lower epidermis helps in the loss of water vapor also. Okay, the water vapor means water, excess water also has to go out. That is also taking place only in this area. The next part, okay, the middle area, if you see the entire area, now it is called as mesophyll. Okay, the entire area is now called as mesophyll. The mesophyll comprises of two parenchyma. One is called as palisade parenchyma and the other one is called as spongy parenchyma. Palisade parenchyma and the other one is called as spongy parenchyma. What is this? These two are new terms for you. Palisade parenchyma and spongy parenchyma together is called as mesophyll. Okay, let's see what is this palisade and spongy. Have a look at this one. This is palisade is given for you, and below that you can see the spongy parenchyma. Okay, spongy parenchyma is here. Okay, palisade parenchyma is here, and here you can see these two terms are being given. Just compare the palisade. Look at the shape. You can see the palisade parenchyma. You can see it is just below the upper epidermis. Okay, and you can see they are all elongated, and another one, there is no intercellular spaces. But if you have a look at the next one, the spongy parenchyma, you can see a lot of spaces. See, have a look here. You can see the gaps here and there. Okay, they are the intercellular spaces. Okay, that is the difference between palisade parenchyma and the spongy parenchyma. Here in the palisade parenchyma, they take part in the process of photosynthesis, and spongy parenchyma, they take part in the process of gaseous exchange. I repeat, mesophyll comprises of palisade parenchyma and the spongy parenchyma. Palisade parenchyma, no intercellular spaces. Spongy parenchyma, intercellular spaces are present. The third difference is, here photosynthesis takes place in the palisade parenchyma and gaseous exchange takes place in the spongy parenchyma. Okay, the next part, let's go on with, in the center you can see the vascular bundle. Okay, have a look at this one. Okay, the center, in the middle part you can see the vascular bundle. Vascular bundle is of course concentrated in the midrib area. Where is the midrib present in the leaf? You can see one thick part in the leaf. Okay, you can see the lines in the leaf, the thick part, the middle part which separates the leaf into two halves, that area we call it as the midrib area. Word itself is the midrib, middle, you can see the rib like structure, so we call it as midrib area. In that area you can see the vascular bundle. Let's see how the vascular bundle is. It is again conjoined, collateral and closed. Okay, closed means here cambium is absent. Again, recollect the terms for conjoint and collateral. Conjoint means if xylem and phloem are present in the same radius, we call it as conjoint, and if the xylem is lying towards the center and phloem is towards the periphery, we use the word as collateral. Okay, please have a look at the diagram. You can see the larger cells of the xylem and the smaller cells of the xylem, also the protoxylem and the metaxylem. You can see. If you have any doubt, just go on with the first slide which we have already learned, where you can see about the protoxylem and the metaxylem. Okay children, is that clear for you? Next we are going on with the next term, which is going to be the bundle sheath. Bundle sheath are nothing but the sclerenchyma cells which is going to be present around the vascular bundle. Okay, that is called as the bundle sheath. Okay, you can see a layer of bundle sheath in your diagram in the vascular bundle. Here the function of the bundle sheath is to store the food grains. Okay, here you can see the bundle sheath here. Okay, so it is just surrounding the vascular bundle. Let's see how the xylem and phloem is going to look like. Okay, xylem is facing the upper epidermis and phloem is facing the lower epidermis. Is it clear for you all? Okay, the notes parts is being given for you, whereby I have given for you the upper epidermis, which is going to be the outermost layer, a single layer without intercellular spaces. Lower epidermis, which is a single layer of parenchyma with numerous stomata. Mesophyll we differentiate into two layers, the palisade parenchyma and the spongy parenchyma. Palisade parenchyma, more chloroplasts are present, and there is no intercellular space, whereas spongy parenchyma, intercellular spaces are present and they help in the process of gaseous exchange. Next is going to be about the vascular bundle, which is conjoined, collateral and closed. Bundle sheath, vascular bundle is surrounded by parenchyma cells called bundle sheath. Xylem is facing towards the epidermis, towards the upper epidermis, and phloem is facing towards the lower epidermis. Okay, now let's go on with the next one, which is going to be the TS of monocot leaf. Okay, now just have a comparison of what we learnt in the dicot leaf and the monocot leaf. In the dicot leaf, we were able to see only one bundle of vascular bundle, okay, whereas here you can see many bundles of vascular bundle in the diagram. In the central part you can see 3 circles, which is going to be red in nature, which is going to be the vascular bundle. Apart from that, the same thing like the similar to monocot, you can see similar to dicot, you can see the upper epidermis and the lower epidermis, but there is slight difference in this one also. Okay, let's have a look at what is going to be there. In the case of upper epidermis, here you can see it is again a single layer which is made up of parenchyma cells. Okay, cuticle is present on both the outer walls, stomata is present in the upper and the lower walls, and apart from that you have got one special cell that is called as bulliform cells. Have a look at this one, children. Okay, you can see in the diagram the bulliform cells. Okay, have a look at this. The bulliform cells are there. They are nothing but the cells in the upper epidermis, whereby some cells become large and thin walled. I repeat, some cells in the upper epidermis becomes large and thin walled. Such cells are called as bulliform cells. Next is going to be, of course, the same, just like the dicot leaf, which is going to be the middle layer, which comprises the mesophyll layer. Let's see what is the mesophyll here. In the case of dicot leaf, we said that mesophyll layer will be divided into palisade parenchyma and spongy parenchyma. Here nothing like that, no such difference at all. Okay, that's an important point here. Mesophyll is not differentiated into palisade and spongy parenchyma. Okay, they are irregularly arranged with intercellular spaces and they contain more number of chloroplasts. That's why it is given green in color. Next, going on with the vascular bundle, let's see how the vascular bundle is going to look like. Okay, each vascular bundle is surrounded by a layer of parenchymatous bundle sheath. Of course, that's the area where it stores the food materials. Okay, next let's see how the vascular bundle, same just like the dicot leaf only. Here also it is conjoined, collateral and closed. Okay, I repeat, conjoined, collateral and closed. Okay, xylem is also similar. Xylem is present, which is facing the upper epidermis, and the phloem is facing the lower epidermis. Have a look at both the diagrams here. Only when you compare both the diagrams, you will be able to see this one. Okay, now let's see how it is going to look like. Okay, the notes part is being given for you. Whereby the epidermis, which is going to be the upper and the lower epidermis. Some cells of the upper epidermis are large and thin walled, called bulliform cells. Mesophyll is not differentiated into palisade and spongy parenchyma. Intercellular spaces are present. Vascular bundles are conjoined, collateral and closed. Xylem is towards the upper epidermis and phloem is towards the lower epidermis. Okay, these diagrams that are being given for you, so far if you have seen in your slides, you would have going with the first one will be regarding the vascular bundles. This is one detailed question, children. You need to practice all the diagrams under types of vascular bundle. That was the first diagram for you. That is an often repeated question. Apart from that, dicot root, monocot root, dicot stem, monocot stem, dicot leaf, monocot leaf. I repeat, these 6 diagrams. Okay, dicot root, dicot stem, dicot leaf, monocot leaf. Of these 6 diagrams, you need not practice all the diagrams. I will give you some important diagrams alone. Only you learn dicot root. So far 6 diagrams is being given for you, of which learn only dicot root and dicot stem. I repeat, dicot root and dicot stem alone, because that is there for your practical part. There for your practicals you have to draw the diagram for your practicals, okay, and it will become very difficult. Since time is there right now, you can practice those diagrams at home with all the labelings for you, okay, and need not practice the dicot leaf or monocot leaf. Time being, you can just give or skip over the diagrams, but go on learning with the notes part. Once the school reopens, we will help you to how to draw those diagrams also. Next in the table you have a comparison between just a small comparison only, the differences between dicot and monocot leaf for you, okay. If you have this one, this is a 2-mark question. Once you get a textbook, children, please mark. I am going on repeating, this is a 5-mark question, this is a 2-mark question. So here and there if you mark, it will become very easy for you. Finally we won't have time to be going on marking all those things. So once you get your textbook, please mark it. I am just marking all the interiors for you. So have a look at all those things. So wherever I have given, please mark it up, okay. The difference between the dicot and monocot leaf. First point, only 2 points is given, it is just for a 2-mark question. It is dorsiventral leaf, okay. The word dorsi means, which is going to be dorsal and the ventral. Dorsal means upper and the ventral, both are the parts are flat. In the monocot leaf, it is isobilateral leaf. Iso means similar, bi means 2, laterals means on the sides, okay, that is the meaning behind it, okay. Isobilateral leaf, you have to pronounce it that way. Split a word and read isobilateral leaf, okay. The second point, the mesophyll is differentiated into palisade and spongy parenchyma. In the case of monocot leaf, mesophyll is not differentiated into palisade and spongy parenchyma. It is an important 2-mark question which you need to read. So as you go, please read and write all the difference between that is being given for you so far. So it will be of use for you while you go on learning in your future. Is that clear for you all children? Okay, thank you children.